7.
Thank you very much for the introduction and the opportunity to be able to talk here. This project is very new, so I'm going to give a brief, slightly elaborate background, followed with the technical details of where we are with the project. And before I start, I just wanted to warn, it's high on biology. So I hope the computer systems here are not going in a ziffy. So, I just wanted to go through the need and the challenge and explain like every element of this so that it is understandable what is the background to that work. So induced pluripotent stem cells can provide effective solutions for cell therapies and regenerative medicine. What is this induced pluripotent stem cells? It is basically the reprogramming of any cell which can divide in the body into a stem cell. And this technology was pioneered by Dr. Shinya Yamanaka. He made a groundbreaking discovery that won him the Nobel Prize in Physiology. So he found a new way to reprogram the adult specialized cells and then turn them into stem cells. Naturally, embryonic stem cells have got pluripotency, which means the cell has the potential to divide to differentiate into any specialized cells. And his discovery meant, theoretically, any dividing cell in the body could be turned into a stem cell with pluripotency. So this actually revolutionized the areas of cell therapy, regenerative medicine, stratified medicine. So as you can see here, all of these areas would have a huge benefit from this technology. So the next statement is that these cells are adherent cell culture systems, which means they can grow and proliferate. For that, they require protein substrates that are compatible with these cells. And traditionally, these cells are grown on feeder cells, which would be mitotically inactivated fibroblast cells or protein extracts, which are again very complex matrices. And in this field, there is a real lack of synthetic materials, which would allow the adhesion and proliferation of these cells so that there is a standardized protocol or method to produce clinically grade cells. So at NPL, we used the technology of self-assembling peptides where you've got, for example, you produce in our case, alpha helical peptide with sticky ends that coil into a protofibril and then laterally associates to form a fibrous structure. And this fibrous structure has got a functional entity where it tethers the Mickey Mouse. So the idea is to take the elements of biology and introduce engineering control. So these matrices, then we went on to test with different primary cells and also human embryonic stem cells. So the work that we did with human embryonic stem cells was very promising. We did this work under the remit of MRC funding with University of Sheffield, their Stem Cell Center. And we found that our matrices are allowing the proliferation and pluripotency of these cells. So we did an extensive qualitative and quantitative work where we looked at pluripotency markers. Now, why is in this field pluripotency is important? That if you've got a substrate which makes the cells to differentiate in a certain way, then those cells lose the potential of being able to differentiate into different types of cells. So in the field, there is a real need for the cells to have a synthetic substrate which will allow the cells to grow and also maintain pluripotency. So this was was very exciting for us because our substrates was allowing the proliferation and pluripotency of these cells. And so we thought that we will put this test, this matrix, through the standardized CCQM process. So this, the stability of this material is tested under another remit, which is VAMAS, and we tested the stability of this material by involving 25 labs internationally, and we found that this material is quite stable. Even after 20th month, we are able to probe the 3 parameters, D1, D2, and the pitch, and we find that we are able to have a very stable material at a nanoscale level. So the last statement is that the quantification of the number of these cells in their proliferative state is an essential step in the development of standardized stem cell culture to realize the therapeutic benefits of these cells. So as you can remember that the normal cells are taken and they are reprogrammed into a pluripotent stem cell. And then at this stage, if you wanted to take this into clinics, you would like to have a standardized way of having enough cells which could be taken into the clinical pathway or the route. And the most straightforward quantification parameters for cell is numerical counting, where 1 can be considered an SI unit. So there are different ways in which we count the cells. We use microscopy, flow cytometer. These are the direct methods. And there are also a lot of indirect methods, for example, which are metabolic based. So in this study, we homed in on microscopy to use microscopy to count the number of cells. So now we come to the nitty-gritty part of the project itself. So the scope of the project, the successful participation in this study will demonstrate the competencies of the labs in differential counting of the stem cells seeded on planar 2D scaffolds and deriving the proliferative stem cell number per unit area and stem cell fraction on planar 2D scaffolds. And the third, documenting the uncertainties of their cell count measurement results. So, just to give an idea of the flow chart for the current study, we needed the 2 materials, the iPSC cells, which we got from NIPS, a well-characterized induced pluripotent stem cells, and we produced the coating material at NPL. And then the next step would be to prepare the standard sample, which will be done in collaboration with INRIM and NIST, and the third would be the data acquisition and analysis. So at the moment, we are in the second step of this study. So to prepare a standard sample, we needed a microscope slide format, and we chose this 8-well chamber, which had a grid in the middle. So the bigger square had a smaller indexed, smaller grids, and those were the small grids would be where the cells will be seeded and would be counted. So we also wanted to know, of course, which was an important parameter, was the area of these small grids. So if you look at the red crossbar, those were the grids that were analyzed for the area at INRIM to get the quantification of the area of these grids. So the next step was to look at the fluorescent biomarkers that could be used in the study. So we chose nuclei stain that would give us, there are quite a few nuclei stain, but we chose 2 to test, which was Hirsch and DAPI. These 2 are quite well-used nuclei stain. And we found both of them were very comparable, but Hirsch was a notch better. So we homed in on Hirsch to use in this study. And in terms of proliferation markers, again, there are quite a few fluorescent markers that you could use, and we tested, for example, Ki67 in a conjugated and unconjugated formats, and ClickIt plus EDU Alexa Fluor 488, and we found that the EDU complex was much more stable to be used. And you can see the excitation and emission detail spectra of these two dyes. So the Hirsch nuclei stain would give us the total number of the cells, and the EDU stain would give us the proliferative cell count. So these two would be used to do the differential counting of the cells to ascertain the proliferative status of the cell. The next very important step was to do the cell seeding density optimization. So we chose a whole range of cell seeding densities, and we analyzed to see what would be the most optimum cell seeding density. As you can see on your left, the seeding density was too high, and the nuclei were overlapping one on top of each other, and it would not make a suitable, it would not be suitable to be counted. Whereas on the right-hand side, you have a density which would be suitable for counting these cells. And this was actually, the background work that was done in P123 was the inspiration behind doing this study to be able to optimize this step. So this is where we are at the moment. The next step would be to have the microscopic setting guidance, which we will do with the help of NIST, and get a really prescriptive guidance to be sent with the standard sample protocol to the participants. So the next, the measurements, very important for this group here. So the proliferative stem cell number per unit area on each substrate will be assessed by counting the number of dual stained cells in the grids that I showed you, which will be in millimeters square. And the proliferative stem cell fraction on each of these substrates will be derived from counting the number of dual stained cells and dividing that by the cells which are stained by Hirsch, which is the nuclei stain, and which will give you the total number of the cell. This is a very busy slide, but I just thought that I wanted to show what was our thought process, for this project. So we are doing this cell number per unit area now, but we would like to also do cell unit per unit volume. That is tricky because you need a 3D system to do that, and that is more physiologically relevant. That is where the cells are in your body in a 3D status. They're not on a 2D status. So we would like to do this and then move on to a cell number per unit volume. It would be quite tricky, but we are developing hydrogels which can be put through this system. And that would then again take you to the measurement services and etc. I would like to summarize that this study is a couple study with VAMAS COG study, where VAMAS we are looking at the material properties, the reproducibility, etc. of the material, and in COG we are looking at the proliferation of induced pluripotent stem cells that are attached and are proliferating on a scaffold material. I would like to acknowledge the NPL biometrology team, all of the COG members, all Laoshi of UK Stem Cell Bank for providing the cells, and the organizing NMIs, NIPS, INRIM, and NIST. Funding body, very important, BASE, and the Stem Cell Center of Stem Cell Biology in Sheffield, and the medical research grant that we got for the stem cell evaluation work that we've done. Thank you very much for the attention.